In chapter 15, we're going to switch gears and stop talking about the human part of infection and instead talk about the microbial part of infection in a process that's called pathogenicity, which in a lot of textbooks is called microbial pathophysiology. Either way, what we're going to be talking about today is how infectious disease takes hold in a body and how it results in infection. Pathogenesis is a multi-step process, as you can see from the flow chart, which begins with exposure to a pathogen, which was discussed in chapter 14. Remember, there are multiple ways that a disease can be transmitted. We have contact transmission, vehicle transmission, vector transmission, and even under those three categories, within contact, you have direct, indirect, and droplet. Within vehicle, you have water, air, and food. And then in vectors, remember, we have biological vectors, which bite to transmit an organism, and mechanical vectors who provide incidental exposure. So there has to be some sort of exposure via transmission from an infected source. And once you've been exposed, that doesn't automatically guarantee that you're going to get sick, right? Remember, this is multi-step from exposure to disease. So the second step in that parade is for the microorganism to be able to attach to not only any part of the body, but actually the proper part of the body. And the reason that part of the body is important is because adherence to your body occurs when the pathogen binds physically to receptors on the cell surface. So in the diagram, you can see these adhesions, which are receptor binding molecules called ligands on the surface of this pathogenic Vibrio cell, and those ligands are binding particularly to a particular receptor on the surface of the cell. And that's something that we discussed with viruses in chapter 13, but that's also true of lots of bacteria. They don't simply lie there, they actually attach to something on the cell because, as you might remember from chapter 16, you are initiating all kinds of innate responses to try to get rid of them. If this happens to be the intestinal epithelium, you're going to try to wash that out by increasing intestinal motility. If this is the epithelium of the respiratory tract, you might start sneezing or start increasing your nasal secretions. If it's your eyes, you might start tearing. So they're trying to fight against that by binding to receptors. And those receptors are found on only particular kinds of cells. Next to that diagram, you can actually see a picture of E. coli, which have been colored yellow, attaching to the surface of the urinary bladder. So exposure via transmission, attachment to the proper body part, and then establishment of infection. Remember, infection is the colonization of the body by microorganisms. So those few pathogens that have landed through transmission to the right place and managed to stay there have to be able to grow there. And as they grow, they will establish a larger infectious process, and that can sometimes be aided by several different kinds of enzymes, which we're going to be talking about later in this chapter. And ultimately, all of those things together produce a disease state, which you'll remember from our chapter 14 terminology means an altered body state. And that disease state can sometimes happen directly through damage by the microorganisms. It can happen because the microorganisms produce toxins, and it can also happen because the immune system over-responds to the pathogen. So there are several different things that come into play when you're trying to figure out what is causing the disease process that describes what illness with a particular microorganism looks like. So in this chapter, we're going to be looking at different aspects of pathogenicity and seeing what kinds of things are involved from the pathogen's perspective in making sure each of these steps is met. The most common portal of entry is the respiratory tract, which is shown on the left-hand side of the screen. We talk about upper respiratory tract infections, URIs, and we talk about lower respiratory tract infections, LRIs. And upper respiratory tract infections are incredibly common, and one of the things that we need to be careful if we contract an upper respiratory tract infection, of course, is that we don't contract a lower respiratory tract infection as a result of that entrance into the respiratory tract. So that's the most common preferred portal of entry, meaning that is the most common place for which microbes have the ability to attach and adhere. Coming in a close second is the gastrointestinal tract. So that would be typically vehicle transmission, contaminated food or water are common gastrointestinal routes of entry, followed by the urinary tract, the parenteral tract, which means your skin, which is why you can see this person's colored a different color and you can see their hair. So that's your skin route. Remember from chapter 16, intact skin is your best defense against microorganisms. So the parenteral route typically happens when the skin has been damaged, although you might remember from our diversity unit, hookworms like Strongyloides are capable of burrowing through intact skin in order to get into the body. And then, of course, at the end there we have the reproductive tract. So these would be sexually transmitted infections, which are direct contact transmitted. And when you're thinking about portals of entry, it's helpful, I think, to think about what kind of disease could get to each of these body systems. For instance, I just mentioned the reproductive tract. So really the only way that things get into the reproductive tract is through direct contact transmission. We talked about parenteral entry being through broken skin, maybe burns or diabetic ulcers or things like that. Respiratory tract, usually contact, possibly airborne transmission. Gastrointestinal tract, vehicle transmission. And thinking about that, also thinking about the fact that for most microbes, the route in, the portal of entry, is the same as the portal of exit. If it goes in through the reproductive tract, it goes out through the reproductive tract. If it goes in the gastrointestinal tract, it goes out the gastrointestinal tract. In the respiratory tract, out the respiratory tract. One really interesting exception to that rule are viruses that include chickenpox and smallpox and things like that, which actually are inhaled through the respiratory tract and then released in pustules on the skin. So it's a different portal of exit than it is a portal of entry. So you actually contract chickenpox by inhaling airborne viruses when a person scratches or moves and disturbs a non-scabbed over pustule. All right, so that gets us through step 1 and step 2. You've been exposed, it's attached to the proper place, but don't worry. There are still other things in place in the body to protect you from getting from exposure to disease. So you can be exposed, you can be inoculated, you can even be inoculated into your preferred portal of entry. And entry does not equal disease, and entry into the preferred portal of entry does not equal disease. A threshold number of organisms have to be reached in order for those organisms to be able to take hold and cause infection, and to be able to move from infection to such a serious disease process that those microbes cause death. And we have 2 terms that are used to describe those threshold numbers. The first, the ID50, describes the infectious dose for 50% of a population. So it's a statistical test that's done to determine how many microbes are going to infect 50% of a test population. And that allows us to compare very different diseases. For instance, inhalation anthrax, which is one of those that prefers the respiratory tract, has an ID50 of about 10,000 spores, whereas Vibrio cholerae, which is a gastrointestinal pathogen, has an infectious dose of 1 million cells. So if you were comparing these two diseases, you might have a difficult time comparing them by severity of disease or route of infection, but you could talk about which of those is more infectious, and that's where we get a difference between communicable diseases and contagious diseases. Contagious diseases, you'll remember from chapter 14, are very easily communicated, and one of the things that makes that happen is having a lower infectious dose. A lower ID50 means a more infectious microbe. So of the two of these, inhalation anthrax is the more infectious than Vibrio cholera. Now, that doesn't mean that it's easier for you to contract. You might remember from our discussion of cholera that cholera tend to have an incredibly high number in the environment, and they persist in the environment for a very long time. And a person who has cholera can excrete millions of cholera cells in every bowel movement. So what it's really telling us is how infectious something is, right? ID50, infectious dose for 50% of the population. Now, what that number doesn't tell us is how deadly either of those infections is. In order to determine how deadly an infection is, we talk about the lethal dose for 50% of the population. So how much is required in order to kill 50% of the population? And again, we can do some comparisons. So at the bottom there, I've got some data, not that you need to memorize any of the data. You should understand what an ID50 is and an LD50 is, but just to give you an idea. So with anthrax, you can actually have a couple of different kinds of infection. You can have cutaneous anthrax, you can have inhalation anthrax, you can also contract anthrax gastrointestinally. And notice, in order to get those infections, there's a very different ID50. So to get cutaneous anthrax, you need an ID50 of only 10 to 50 spores. The respiratory tract, as we mentioned earlier, 10,000. The gastrointestinal tract, 250,000 to 1 million is the ID50. So far easier to contract it from the skin. However, skin infection with anthrax is not nearly as lethal as inhalation anthrax. Now, moving over to the LD50, we're looking here at 3 different kinds of toxins, which are something we're going to talk about in a few minutes. One comes from certain strains of Clostridium botulinum, and it is called the botulinum toxin, and it has an LD50 of 0.03 nanograms per kilogram of body weight. Shiga toxin, which is found in different enteric bacteria, has an LD50 of 250 nanograms per kilogram of body weight, and staphylococcal enterotoxin has an LD50 of 1,300 nanograms per kilogram of body weight. So when you look at those 3 numbers, you want to ask yourself, which of the 3 of those is more deadly? And the answer to that is botulinum toxin, which in fact is the most potent toxin known to man. And just to give you a point of reference for that 0.03 nanograms per kilogram of body weight, if you had a milligram of botulinum toxin, you could kill 1 million guinea pigs with that. So we're building this idea of exposure to disease. Now we understand you have to be exposed, it has to adhere to the right place, and it has to get there in sufficient number to cause infection. And then the next step in the parade is to invade into the body, which I mentioned earlier, is helped along sometimes by enzymes. Now, there are dozens of microbial enzymes that we could talk about. We are going to talk about 5, and you can see those 5 here, and they are hyaluronidase, collagenase, coagulase, kinase, and leukocidins. Those of you who had taken anatomy and physiology might recognize parts of hyaluronidase and collagenase because you know that collagen and hyaluronic acid are connective components of the epithelium. You can see in the picture there, you have a collagen layer underneath the epithelial cells, and sort of the cement that holds those cells together is hyaluronic acid. So if you have an enzyme-producing bacterium that is capable of producing hyaluronidase because it has the gene for hyaluronidase. That hyaluronidase will be excreted by the bacterium as an exoenzyme, and it will digest the hyaluronic acid and allow it to penetrate the epithelium, getting through that first and primary layer of the body's defenses. Some cells further invade the body by producing collagenase that allows them to digest the collagen and get into the lower layers of the skin. Now, I want you to be thinking, when you're thinking about this, also thinking about what the body's doing. So this is damage to cells, and damage to cells results in cells releasing chemicals that initiate the inflammatory process. And all of the different things that we talked about in chapter 16 that happen as a result of inflammation happen here. You have recruitment of white blood cells, you have dilation of blood vessels, you have increased space between the cells of the capillaries, allowing for fluid to leak into them. And so from the bacterium's perspective, it needs to be able to deal with the threat that is coming, and that is the white blood cells. So sometimes these cells can also produce an enzyme called coagulase, and coagulase allows them to take the clotting proteins that have been released from the capillary into the interstitial space to form a clot around themselves, which may seem a little strange, but what that clot will do is hide them from the white blood cells that are going to be coming in response to inflammation. So coagulase is a highly pathogenic enzyme because it hides these microorganisms from the white blood cells. The white blood cells come in, they notice the clot, they think that that's something that's already been taken care of, and they ignore it. And if inflammation starts to subside, they go away, and now those microorganisms, which are hiding inside of the clot, can produce a second kind of enzyme called kinase. We have staphylokineses, we have streptokinases, and those kinases dissolve the clots and allow those cells to release themselves from that clot and get into the body and cause further damage. So those are really helpful enzymes both for establishing infection and allowing invasion, moving from local infection to systemic infection because you've made it into the bloodstream and then possibly through the bloodstream making their way to a different particular part of the body. Now let's say that you don't produce coagulase and you can't hide yourself from white blood cells. So the white blood cells come in as a result of inflammation. They engulf you through the process of phagocytosis, and you think, oh no, all is lost, but that's actually not the case because some of these bacteria are capable of producing leukocidins, which, as their name suggests, are capable of killing white blood cells. So the white blood cell engulfs these bacteria and they actually blow it up from the inside out. So what was supposed to inactivate the threat has actually died as a result of trying to do that work. So enzymes allow bacteria to penetrate the body, to get through the host defenses, and then help them to fight off all of these innate defenses that are coming on board to try to rein in the infection before it can get established. One other thing that we want to talk about in terms of increasing pathogenicity of bacteria are toxins. And toxins are a catch-all phrase for several different kinds of things, but when we talk about toxins in the microbial world, we're talking about toxins that are produced by bacteria, which are able to cause systemic damage to the host, even though the cell itself might be kept in a local area. We talked a little bit about some of this terminology. We've heard the word toxemia before, and you'll remember emia always refers to blood, so toxemia is toxins in the blood. We talked in chapter 18 about toxoids, which are immunizations against toxins rather than immunizations against the bacterial host, and that includes things like pertussis and diphtheria and tetanus, where it's not the cell that's dangerous, it's the toxin that the cell produces that's dangerous. So immunizations against toxins can be more helpful than immunizations against the cells. We also talked in chapter 18 about a case study where a little boy hit his head and got tetanus toxin And part of the therapy to help him to get over that was to be given passive immunity artificially by giving him antibodies to that tetanus toxin. So we've seen some terminology. Now we're going to introduce two more terms, exotoxin and endotoxin. Exotoxins are genes that are transcribed and translated into toxins that are excreted from the cell, and they're found commonly in gram-positives, but they are also found in gram-negatives because they are simply part of the chromosome. Endotoxins, on the other hand, are always and only found in gram-negative cells because they're actually part of the gram-negative lipopolysaccharide outer membrane. So whereas exotoxins are genes that are transcribed and translated into these proteins which are excreted, and that's why they're called exotoxins, endotoxins are part of the bacterial cell, and they are released only when the cell ruptures as a result of its death. We're going to spend quite a bit of time talking about different kinds of exotoxins because there happens to be a lot more variety in them, and then we'll wrap it up by talking about endotoxins. So just to give it a bit of context, we have three different types of exotoxins. Thankfully, they're called type 1, type 2, and type 3. We're going to look at an example of each of those. All of type 1 are called type 1 because they are cell surface active, so they bind to the surface of a host cell and have some effect on that cell. Type 2 are membrane damaging, and type 3 have an intracellular action, so either acting on the cell surface, damaging the surface of the cell, or having some kind of intracellular action. And then endotoxins, for the most part, cause fever, but we will see one endotoxin toward the end of this chapter that has a particularly devastating systemic effect. So the example of a type 1 exotoxin that we are going to be looking at is the superantigens, which cause toxic shock syndrome. What happens in toxic shock syndrome is something called a cytokine storm, where you have this unbelievable activation of T cells, far and away more than needs to be happening, and that kicks off a terrible string of events which can lead to death, and in the case of toxic shock syndrome does. So here in the diagram you see a T cell who's coming in to see his antigen presenting cell, right? This is a CD4 positive T cell, which means he's a helper T cell. He's looking at this antigen presenting cell who's presenting an antigen to him which he would not normally recognize because that's not his antigen. But the presence of this super antigen, which was a gene transcribed and translated in, say, Staphylococcus aureus or Streptococcus pyogenes, bridges that gap and causes this helper T cell to think that he's been activated. And so he will undergo clonal expansion and differentiation into effector cells, and he will go out and he will turn on the rest of the immune system. So rather than just the appropriate T cells getting activated, at least half of the T cells in the body are activated. And this begins with fever and muscle aches and headaches and chills, and it may include nausea and vomiting and diarrhea, and it moves fairly quickly to low blood pressure, high heart rate, rapid breathing, and high fever, and it ultimately ends with circulatory shock, multi-organ failure, and death. So this super antigen, because it's capable of recruiting far more of the immune system, is actually making the body damage itself. So you'll remember from the beginning of the chapter I said that sometimes what happens to damage us is not caused directly by the microorganisms. They cause us to hurt ourselves. And that's an example there with a type 1 exotoxin, which, remember, is a cell surface active exotoxin. So it's just acting on that receptor of the T cell and activating it. Another type 1 exotoxin which acts on the cell surface is the neurotoxin botulinum toxin, which is produced by Clostridium botulinum. And C. botulinum produces a toxin which interferes with neurons. It doesn't kill the neuron because it's just a cell surface active toxin, but what it does is it changes the way a neuron interacts with the muscle that it's innervating. So in a normal neuromuscular junction, you have neurons that carry with them impulses from the central nervous system, which are going to communicate to the muscle that the muscle should contract. And it does that by releasing a neurotransmitter called acetylcholine, which is listed there as little capital A's. Those A's are released from the neuron. They attach to the muscle, and that kicks off a series of events which results in muscular contraction. So the brain says contract, the muscle contracts. Well, botulinum toxin, when you get botulism, which is a type of food poisoning typically, the botulinum toxin binds to the end of the neuron and blocks the release of acetylcholine, and the muscle never contracts, and you end up with the form of flaccid paralysis. And what will ultimately happen in a person who has botulism food poisoning is they'll begin with difficulty swallowing and things like that. Ultimately, though, this botulism toxin will find its way to the respiratory muscles and will cause respiratory paralysis, and the person will suffocate. So you have to put them on supportive therapy in order to breathe for them until these toxins can be bound up by the body and removed from the body. Incidentally, we talked about Clostridium tetani, which has a similar mode of action, but what it does is blocks a neurotransmitter that would allow a muscle to relax. And so with tetanus, you'll remember our case study, the boy had spastic paralysis. He had muscles that were unable to relax. And remember, he also needed mechanical ventilation, because whether or not your respiratory muscles are paralyzed in a contracted state or paralyzed in a relaxed state, you can't breathe, you can't exchange gases, and you suffocate. So whether you have flaccid paralysis or spastic paralysis, either of those two conditions would require ventilation. Now, the good news is, because this is a type 1 exotoxin, it's binding to the surface of the cell, but it's not damaging the membrane. If you can get rid of the toxin, you can recover completely from the infection, which is thankfully what happened with the little boy in our case study. Type 2 exotoxins, on the other hand, damage the membrane of cells, which typically results in the cell dying. So we call them cytotoxins because they're cell killers. So type 1 are not cell killers, type 2 are cytotoxins, the ones we're talking about here, and they kill the cells that they target. The one that we're going to use as an example here are a subset of the cytotoxins called hemolysins, and we're mentioning those because those are highly relevant to the differentiation and study of staphylococcal and streptococcal bacteria, because pathogenic strains and species of Streptococcus and Staphylococcus are capable of producing different kinds of hemolysins, right? Streptococcus pyogenes produces streptolysins, and Streptococcus agalactiae produces beta hemolysins. Staphylococcus aureus produces alpha toxins. All of these are hemolysins. They attach to and blow up the red blood cells, and we study those in class by growing microorganisms on blood agar. And when the blood is hemolyzed, the agar turns a yellow color. And of course, that's pathogenic to us because it's blowing up our red blood cells, and it's really helpful to the pathogen because it's liberating iron and other things that they will grab out of the environment once the red blood cell has been destroyed. One other clinically very interesting type 2 exotoxin is streptolysin S, which is produced by group A strep, which are Streptococcus pyogenes, and which can cause NSTIs, which are necrotizing soft tissue infections. And they're really interesting. If you look here, you see all of these little green circles. Those are the streptococcal bacteria, and they're moving from the epidermis into the dermis into the subcutaneous fat because they produce the streptolysin S, which allows them to move into those areas. And when they do, they start binding to neurons, and they shut down the inflammatory response, so they increase neuronal pain. The white blood cells are not recruited into the area. They impair the phagocytes locally, and they're incredibly cytotoxic to keratinocytes, endothelial cells, and neutrophils. And so this streptolysin S, because it's able to do all of these different things, shuts down the immune response, causes hyperinflammation and tissue damage, and it causes these terrible, terrible infections, which happen very, very quickly and can move systemically quickly. The last kind of exotoxin we're going to talk about today is a type 3 toxin, and remember, all type 3 toxins have an intracellular effect, and we're going to be talking about AB toxins, which are a type 3 exotoxin composed of two proteins in a dimer. So there's an A portion and a B portion. The B portion actually binds to the host cell, and that causes the entire complex to be endocytosed into the cell. And once it's inside the cell, the A component will be released and it becomes enzymatic in the cell. So that's how they're able to cause some intracellular issue. And there are all sorts of AB toxins that you could look at. We're going to look at two today. The first of those is the diphtheria toxin, which is found in only pathogenic strains of Corynebacterium diphtheriae. And the way that that toxin works is to bind to different host cells, and it uses that B portion to bind that causes it to be endocytosed, and then the A portion goes in and affects the way that translation of proteins occurs. And because it's able to halt protein synthesis in the cell, it kills the cell. And one of the effects of that is most visible is something called a pseudomembrane that will grow across the surface of the tongue and the roof of the mouth, as shown in this diagram here. And the reason that this disease becomes deadly, which it is not because of this membrane in the mouth. It's because that same kind of membrane grows in different areas of the body, and it tends to move preferentially to the heart or the liver, and it causes necrosis there, and that is what causes death. So necrosis of the heart or liver in diphtheria sufferers will cause death. The final type 3 exotoxin that we're going to mention, and we'll be almost done with exotoxins, is the enterotoxin produced by the gram-negative Vibrio cholerae. So not only gram-positives, but gram-negatives as well can produce these type 3 exotoxins. Again, with Vibrio cholerae, you'll remember that you have that two-component system. The first one binds to the cell, and that allows the second part to come in and affect some portion of the cell's metabolism. And in the case of Vibrio cholerae, you might remember that's a gastrointestinal disease. It was studied by John Snow in London. It still today is a killer of millions of people. And the reason that it kills people is by causing massive dehydration in a very short period of time. And that happens as a result of this toxin produced by certain strains of Vibrio cholerae. So it binds to the cells in the intestinal epithelium, and without going into too much detail, it blocks the absorption of sodium from the lumen, the space of the intestines, which, as you can see in step 3, then causes the movement of chloride ions out of the body into the space of the intestines to try to neutralize that positive charge. And once you have salt in the space of the intestine, then the body will actually lose water as that water moves osmotically into the space of the intestines in order to dilute the salt. So with this kind of diarrhea, you don't just have passive diarrhea where you're not absorbing water from the intestinal tract because you're moving things through too quickly. You have that, and you have active movement of water from the body into the space of the intestines. And people with cholera can end up losing 20 liters of fluid per day, and you can die within 24 to 48 hours if you don't have oral rehydration therapy. All right, one really cool thing that's been discovered after people understood how these AB toxin works is that they could be used to target and kill particular cells by targeting particular cell receptors. So recently there was a phase 1 clinical trial on minimal residual hairy cell leukemia, which is a mature B cell neoplasm in which the CD22 receptor is upregulated. So these cancerous cells produce tons and tons of these receptors and have them on the surface of the cell. So this immunotoxin is made to bind to the CD22 receptor and then become endocytosed, which liberates the enzymatic portion of the toxin, which moves through the cell, inhibits protein synthesis, and accomplishes a programmed cell death called apoptosis. And overall, the positive response in patients to this therapy was 88%, with 64% of them going into complete remission. So the targeting of particular cells with this immunotoxin, which is a modified AB exotoxin, provides a much more pinpoint response than something like chemotherapy, which tends to have drastic side effects because of the fact that it affects multiple cell types within the body. And finally, as promised, endotoxins, which are nice, right? So endotoxins primarily cause fever. And remember, they're called endotoxin because they are they are inside of the cell. They are part of the lipopolysaccharide membrane, which, because it's only found in gram-negative cells, means that endotoxins are only found in gram-negative cells. So when endotoxin-containing cells, which are gram-negative, are engulfed by a macrophage, or a dendritic cell, which is the job of the macrophage and the dendritic cell, that bacterium will be transported over to a lysosome, and the phagosome containing the bacterium will fuse with the lysosome, and that will cause the digestion of the bacterium into all of its little parts, which include the endotoxins. And once those endotoxins are liberated, they cause the bacterium to release a chemical messenger called a cytokine, which moves through the blood vessel into the brain and causes the hypothalamus to produce prostaglandin, which causes fever. Now, this may sound familiar to you because we talked in chapter 16 about the fact that these cells, the macrophages and dendritic cells, are capable of causing fever as a result simply of their engulfing bacteria. So what this means is that this is fever on top of the normal fever that would happen. So this is basically the difference between low-grade fever, which is good, body's fighting, to high fever, which is damaging because you have a massive release of these cytokines and an ever-increasing temperature in the body. And so a lot of times that's a clinical marker between bacterial and viral diseases. Bacterial diseases are far more likely in the absence of other symptoms when the fever is above 105. When the fever is below 105, it's much more likely, absent other symptoms or signs, that you have a viral infection. So endotoxins primarily cause fever. We're going to talk about one exception to that now, but for the most part, we're just going to say endotoxins cause fever. So one endotoxin we want to talk about for just a few moments is the endotoxin produced by Neisseria meningitidis, and whose pathophysiology begins with damage to endothelial cells, but can result in all sorts of damage and ultimately death. So this starts when the endotoxin binds to the endotoxin binding protein of macrophages and dendritic cells, and it causes an intense inflammatory process to be triggered, which causes the endothelium, the cells of the blood vessels, to become sticky. And because they're sticky, you get pore clotting elsewhere, So you get this disseminated intravascular coagulation where you have weird little clots forming in small blood vessels, and that depletes platelets and clotting factors, which means that you get excessive bleeding elsewhere. And when you get that excessive bleeding and the damage, you get these little tiny red spots under the skin, which are called petechial hemorrhage. And one of the classic signs of Neisseria meningitidis infection is the presence of these little red areas on the skin, which if you push on them remain red. And that's can be seen in the diagram here where you've pushed a glass up against the skin and the redness stays. So we call that a non-blanching rash, and that's a result of a petechial hemorrhage, a hemorrhage under the skin of tiny blood vessels. So that's actually bleeding under the skin. It's a result of this sticky endothelium and the damage to that blood vessel. Once you have damage to enough of those blood vessels, of course, that means that the tissue is not fed and the tissue dies. So you get peripheral necrosis as a result of these bleeding episodes. You'll also get severe myocardial dysfunction, shock, multi-organ failure, and this can very quickly lead to death. So anytime fever with this petechial rash is noted, that's always evidence that somebody needs to go to the hospital and get treated very quickly, because this meningitis septicemia, even if it doesn't go into the central nervous system, as its name suggests to you that it might, right? Meningitis, meningitis, but even meningococcal septicemia causes all of this damage and can kill you. Multiple stories have been told of people who don't feel well, go to sleep, wake up, or found with this rash and end up waking up in the hospital, days, weeks later, and they've had limbs amputated as a result of this infection. So most endotoxins cause fever, but there are these exceptions to that rule, one of them being the endotoxin produced by Neisseria meningitidis, which causes meningococcal septicemia. Now, we haven't talked a lot about viruses in this chapter, and the simple reason for that is the chapter isn't large enough really for us to talk about all of the things that can happen and do happen. So we focused primarily on bacterial diseases. And one of the things that will allow us to talk about viruses when talking about increased pathogenicity of bacteria is the fact that bacteriophages are capable of undergoing lysogenic cycles where they attach to bacterial hosts, inject their DNA into the host, and that DNA becomes part of the bacterial chromosome. And a lot of times that DNA, shown here in pink, which is replicated along with the cell every time it undergoes binary fission, can have on it these toxin genes that we've been mentioning throughout the chapter. So notice Vibrio cholera. The strains of Vibrio cholera which produce the disease cholera, because they have the gene for the cholera toxin, have that gene because they themselves are infected with the CTX phase. So Vibrio cholera plus the CTX phase means that you have a cholera toxin producing Vibrio cholerae, which is capable of producing the disease cholera. Likewise, Escherichia coli, when it has been lysogenized by the lambda-phage, gets with that lambda-phage DNA a Shiga-like toxin, which allows it to cause enterohemorrhagic E. coli disease. Corynebacterium diphtheriae, if it's lysogenized with the corynebphage. Beta has now a diphtheria toxin, which causes diphtheria. So we talked about cholera. We talked about enterohemorrhagic E. coli in earlier chapters, and we were looking at how to diagnose different kinds of things with PCR, so that might sound familiar to you. We haven't talked about this next one, Streptococcus pyogenes. We did talk in chapter 18 about the fact that Streptococcus pyogenes can cause scarlet fever, and you might have been wondering, well, why doesn't it always cause scarlet fever? Because only those Streptococcus pyogenes which have been infected themselves with the T12 virus will have the ability to make these erythrogenic toxins which cause scarlet fever. And then finally, Clostridium botulinum can be infected with various different kinds of phages, which bring with them the botulinum toxin and allow him to cause botulism. So there are strains of these bacteria which are toxigenic and cause these diseases, and there are strains of these bacteria which are non-toxigenic and don't cause these diseases. So the enzymes we've talked about can increase pathogenicity, the exotoxins, the endotoxins, the infection of the bacterium with viruses can increase pathogenicity. And so just to sum all of those up, we have a diagram here of a salmonella cell who has a lot of different factors which are increasing its virulence. And in your lecture notes, you actually have a summary table of the mechanisms of pathogenicity as well. So there are enterotoxins, which increase its pathogenicity because they cause diarrhea. There are things called siderophores, which chelate iron or grab iron out of the environment in order to help this bacterium to grow. You'll remember from chapter 16, we talked about one of the benefits of fever being that it hides iron. Well, this allows them to grab iron. This cell also has fimbriae, which we haven't talked about in a while, but fimbriae allow them to attach, right, to adhere. So first step is exposure. Remember, second step of infection is adherence. There are also these surface appendages which help with adherence. There are cytotoxins, which are capable of inhibiting cell protein synthesis, right, like the AB toxins that we talked about before. This cell also has a capsule, which inhibits complement binding. It also makes it more difficult for this cell to be phagocytized. It's also motile via peritrichous flagella, which allow it to swim away from white blood cells and escape your immune system. They also have antigens which allow them to adhere and inhibit phagocyte killing. They have O antigens which inhibit phagocyte killing. They have anti-phagocytic proteins. And finally, they have an endotoxin in the lipopolysaccharide outer membrane, which will cause fever. And that's just on the outside of the cell. Inside of the cell, there's a lysogenized viral gene shown in red, and on top of that, a virulence plasmid, which is going to carry with it resistance to antibiotics. So multiple things can come together in these bacteria in order to create increased levels of virulence. And the thing that you want to remember is most of this is genetically determined, and you'll remember from chapter 8 that bacteria share genetic information with one another very easily in the environment, and that's why we see Shiga-like toxin in E. coli, and we see different kinds of phages causing increased virulence in multiple kinds of bacteria, and we see antibiotic resistance being shared amongst multiple species and genera of bacteria because they share this genetically derived information. So as a recap, pathogenicity, microbial pathophysiology begins with entry, right? And you don't just have to be exposed, you have to be exposed in the right way to the right place, meaning the proper portal of entry, which remember from our discussion earlier in this chapter, the respiratory tract is the most preferred portal of entry. Whether or not you get into the body is not just dependent on the preferred portal of entry, it's dependent on a threshold number of microorganisms, which then can adhere to the cell and increase their pathogenicity by the production of enzymes or toxins, causing increased levels of infection, increased damage to the body, and then finally you become the vector and you excrete that bacterium out into the environment, typically through the same route of exit as was the route of entry.